So today we're going to talk about Mendelian crosses and a dihybrid cross. Um, for this problem, what we're going to look at is when we have two traits that are each governed by their own gene, and each gene only has two alleles, and that these two genes are occurring on separate chromosomes, or we would say are sorting independently. And when we have this sort of situation, we can make some predictions based on the work Mendel did with his peas. And because these two genes are sorting independently, or that they are on separate chromosomes, so during gamete formation, how one gene assorts into the gametes is completely independent of how the other gene does. We can consider these as separate events, and we can calculate our probabilities separately for each gene, and then combine them later on with our rules of probability. So remember our rules of probability, if we can make the statement that the genes, that the one event and another event, and use the word and, we're going to multiply our probabilities. If we can make the statement that it's one event or another event, with the or in our statement, we're going to add our probabilities. So for this first problem, probably one of the more challenging things that people will find when they're working out problems like this is the fact that is trying to convert the terminology that we find in the problem and into identifying the meaningful information that we need. And so that's part of what we're going to do here. So when we look at this problem, it's talking about cats, and that cats could either have curled ears or normal ears, and the cats can either be black or they can be gray. Now, we're told in the problem that the curled ears in the cats results from an allele we're going to call capital E, and we're told that this allele is dominant over the recessive allele, which gives normal ears. So what that means is when we have a cat that has a genotype E with a big E and anything else, that genotype is going to equal curled ears. And if we have a genotype in which we have two recessive alleles, now the line here means that any the line there means that any either a big E or a little e allele could be there, and that's what we mean by dominant. When we have a homozygote recessive cat for the little e, we're going to have normal ears. Okay. Then we're also told in this problem that the gene that's assorting independently, we're going to give call it G, that black color allele for this gene, big G, is dominant over the gray allele, little g. Okay, so if we have big G with anything, we have a black cat, and if we have little g, we have a gray cat. Okay. So now, if we look at the rest of the problem, we're told that we have a gray cat that's homozygous for curled ears. So a gray cat means that in the G gene, they're little g, and then has curled ears, so has at least one big E, and then we're told that it's homozygous for curled ears, so must have two big E's, must be big E, big E. Okay, so for the P generation of this cross is going to be big E, and then, oops, sorry about that. And then when we look at and then we see that this cat is for curled ears, and it's a gray cat, so that means it's little g, and this cat is crossed with a homozygous black cat, so black, homozygous with big G, so big G, and that the cat is has normal ears, so has little e. Okay, so now, and then we're told that in the F1 for all these cats are black and have curled ears. So the F1 generation would be big E, big G, little g. And the reason that I know that is because this parent can only give a big E, so the offspring has to have a big E, and this parent can only give a little e, so it must have a little e. Okay. Likewise, this parent can only give a little g, so all the offspring in the F1 must have a little g, and this parent can only give a big G, so all the offspring must have a big G. So then for the rest of the problem, what we're told is that if two of the F1 cats are mated, what phenotypes and proportions are expected in the F2 generation? So there are two different ways we could go about solving this. There's a really long, drawn-out way, which I'll start to do, but I'm going to stop because I'm going to explain how there's a quick, easy way for us to work this problem out. So what we're going to do is we're going to be taking big E, little e, and big G, little g, one of those F1 cats, and we're going to cross it with another F1 cat. Now these are independently assorting alleles, so we can consider the E gene separate from the G gene, and so we can calculate probabilities for the offspring in the F2 generation independently for the E and the G. Okay, so in order to do that, we can just set up a quick and easy Punnett square to kind of see what our probabilities would be for each of the genotypes. And these are just monohybrid cross of a heterozygote. So what we have is a 3-to-1 phenotypic ratio. And so that 3-to-1 phenotypic ratio, remember that our big E means that those cats have curled ears. So we have a 3-to-1 ratio of curled to normal ears. Okay, but we also have a 1-to-2-to-1 ratio of 1 homozygote dominant, 2 heterozygote, 1 homozygote recessive. Now if we did for G, we're going to get the same exact thing. Big G, big G, little g, big G, little g. We will get, of our 4 possible offspring, we'll get 1 big G, 1 big G, little g, 1 big G, little g, so 2 heterozygotes and 1 homozygote recessive. Now the question is asking, what are the phenotypic proportions that would result from this cross, considering both of the genes and both of the genotypes? Now, in order to do that, we can do one of two things. Now, we can work out our probabilities independently and start pulling them together. So we have and for this one, we have a 1/4 probability of a big E, big E. We have a 1/2 probability of a big E, little e. We have a 1/4 probability of a little e. Okay, and the same thing would go for the G's. We could big G would be 1/4. Big G, little g would be 1/2. Little g would be 1/4. Now, why this gets to be a very long, drawn-out process is because we could say, okay, this big E could meet up with any one of these combinations. So to do the fork line method, what that means is we'd set up big E as 1/4, could meet up with a big G, could meet up with a big G, little g, or it could meet up with a little g. So we would have all of these possibilities, and this would be 1/4, 1/2, and 1/4. So we have the possibility of 1/4 times 1/4 being 1/16 of the time we're going to get big E, big G. 1/8 of the time we're going to get big E, big G, little g. and then 1/4 times 1/4 is 1/16. We're going to get big E, little g. And then for each of these, we want to say what the phenotypes would be. So remember, if we have any of big E's, these are all going to be curled-eared cats. And if we have a big G, it's going to be black cat. So these all would be black for these first two. Black. And then we could say this would be gray. Okay. And then we could do that again. We could just say, all right, we now have a 1/2 probability of it being a big E, little e. And that could meet up with the big G, and the big G, and the little g, and do out this whole thing once again. Now, that's what you would probably do if this wasn't of a particular type of problem that I'd like to point out to you. So remember when Mendel worked with his peas, and we consider a cross where we maybe have round peas and wrinkled peas, and yellow peas and say round and yellow peas with green and wrinkled peas. That was his classic dihybrid cross. When he would do one of these dihybrid crosses, we would get, didn't matter what traits we had, we start off with a homozygote dominant for each of the traits, and a cross with a homozygote recessive for each of the traits in the P generation. The F1 generation are all heterozygotes. We would end up with a characteristic proportion in the F2 generation. And so what I'd like to point out is while when we did round yellow peas and green wrinkled, we had our recessives and our recessives together and our dominants and our dominants. In this case, we have our dominants in one parent paired with a recessive, and we have a dominant paired with a recessive. But the important thing to notice is that in the F1 generation, we have heterozygotes for these two independently assorting genes. So when we cross these two independently assorting genes that have two alleles, we're going to get Mendel's ratio of Mendel's ratio of 9 to 3 to 3 to 1. So we'll end up with a 9 to 3 to 3 to 1 ratio. So the question is what does 9 to 3 to 3 to 1 mean? Well, just like when we looked at a monohybrid cross, and we said, okay, we have a 3 to 1 ratio. What we're talking about are the phenotypes that we find for that particular one trait. And so in this case, we have 3 curled ears to 1 normal ears. Well, 9 to 3 to 3 to 1 is the same thing, only what we're going to find is 9 dominant traits. So remember, the dominant trait is the big E and the big G. So we'd have 9 black curled-eared cats. Then we'd have 3, 3, 1, where 3 is a dominant recessive. The next 3 is recessive dominant, and the last one is recessive recessive. And so in this case, if we did dominant and recessive, we would have a gray cat that has curled ears, gray being recessive. And then sorry about that. curled ears. And then this one would be the reverse of that, where we'd have a black cat being the dominant and normal ears being the recessive, and then the two recessives together, gray and normal. Okay, so that's how we solve that problem. Now, the next problem is similar, but now what we're going to do is we're going to take one of the F1 cats and we're going to cross it with a cat that is homozygous recessive for both of the traits. So we see that the F1 cat is going to mate with a stray cat that's gray and possesses normal ears. So gray means little g, normal ears means little e. And so what type of cross is this? What we want to remember here is this is just a classic type of genetic cross. Whenever we're going to be crossing one organism with a known recessive for particular sets of traits, this is what we refer to as a test cross. Okay, so then what are the phenotypes and proportions for the progeny that are expected? And for this one, doing the Punnett square like I started with the last one is probably the easiest way and the fastest way, because this isn't a standard dihybrid cross. It doesn't have a standard set of proportions. And so what we can see is when we do this is that we have an F1 cat. So the F1 is, remember, heterozygous for both, crossed with a homozygous recessive cat for both traits. And so we can do two Punnett squares for this to kind of see what our ratios for each gene are. And we get 1/2 of our offspring are heterozygous, 1/2 of our offspring are homozygous recessive. So we have 1/2 big E, 1/2 little e, same thing here, 1/2 big G, 1/2 little g. And so when we combine these probabilities, I need to apparently get some more memory. Okay, we could have that reach that one, and that reach that one. Okay, so we have a 1/2 probability of our big E, little e, crossing with our fertilizing our 1/2 big G, little g, which would equal 1/4. They're going to be big E, big G, little g. And then we can keep on doing the same thing for the rest of them. We would have 1/2 big E, little e, crossed with 1/2 little g, which will give us 1/4 big E, little g, and get this out of the way. And same thing again. You have 1/2 little e, crossed with 1/2 big G, little g, equals 1/4 little e, big G, little g. And then last one is the same. We would do 1/2 little e cross with 1/2 little g, which would give us 1/4. So we have a 1 to 1 to 1 to 1 to 1 ratio, or 1/4 to 1/4 to 1/4 1/4 ratio. So a quarter of each of our offspring would have these genotypes. And the last step of solving this problem would be actually assigning phenotypes to these. So these would be black and curled. These would be gray and curled. Sorry about that. Little g would be gray. So we have gray and curled. These would be the little e gives us normal ears. And the big G, little g gives us black cat. And then we have little e, and little g would be a gray, normal eared cat. Okay. So now the key to understanding this is the fact that the genes assort independently. We have 2 genes, 2 alleles, and you could do this with as many genes as you need to, as long as they're sorting independently and you only have 2 alleles. Okay. In which case, then you can calculate each probability separately and then combine them at the end. And so the biggest trick that most people have with solving these sorts of problems is trying to establish what the phenotypes are and what the respective genotypes are. As long as you can work out what the parents are, then you can figure out what the offspring are going to be. Okay, and you can make your predictions. And so we could ask any sort of question we could give you like that last cross and ask you questions like, what's the probability of getting a gray curled-eared cat? And you would say it's 1/4, or that you have a 25% chance. Okay. So that's how you solve Mendelian dihybrid crosses. I hope this video helped. If you want to give me a thumbs up to say yes it did, that would be great. And otherwise, have yourselves a great day.